In the last section, we have discussed the important physiological characteristics of the large intestine. Now we are going to talk about the role of dietary fibers in the large intestine and defecation reflex. We will also shed light on the role of brain and gut microflora in this section. Dietary fiber or roughage is a group of food particles that pass through the stomach and small intestine without being digested and reach the large intestine unchanged. Other nutritive substances of food are digested and absorbed before reaching the large intestine. A characteristic feature of dietary fiber is that it is not hydrolyzed by digestive enzymes, so it escapes digestion in the small intestine and passes to the large intestine. It provides a substrate for microflora of the large intestine and increases the bacterial mass. The anaerobic bacteria, in turn, degrade the fermentable components of the fiber. Thus, in large intestine, some of the components of fiber are broken down and absorbed, and the remaining components are excreted through feces. Now, the question arises, what are the components included in dietary fiber? Major components of dietary fiber are cellulose, hemicelluloses, D-glucans, pectin, lignin, and gums. Cellulose, hemicelluloses, and pectin are partially degradable, while other components are indigestible. Dietary fiber also contains minerals, antioxidants, and other chemicals that are useful for health. One of the most important characteristics of the large intestine is the defecation reflex. When a critical mass of feces is forced into the rectum, the desire for defecation is experienced. This sudden distension of the rectum walls produced by the final mass movement leads to a defecation reflex. This reflex comprises a contraction of the rectum, relaxation of the internal anal sphincter, and initially, contraction of the external anal sphincter. This initial contraction is soon followed by a reflex relaxation of the sphincter, initiated by an increase in the peristaltic activity in the sigmoid colon and pressure in the rectum. The feces are then expelled. This reflex relaxation can be overridden by higher center activity, leading to a voluntary control over the sphincter, which can delay the expulsion of feces. The prolonged distension of the rectum then leads to reverse peristalsis, which empties the rectum into the colon and removes the urge to defecate until the next mass movement and/or more convenient time. The chyme that enters the large intestine is isotonic. However, in the colon more water than electrolytes is absorbed, leading to water being absorbed against a concentration gradient. The process is controlled by sodium-potassium at paces located in the basolateral and lateral membranes of the epithelial cells that line the walls. During the defecation process, the following structures of lumen play a role. The mucosal surface of the large intestine is relatively smooth with no villi, only microvilli. However, crypts are present, and most cells are columnar absorptive cells with a large number of mucus-secreting goblet cells. Sodium is extruded by the membrane pumps into the extracellular spaces. Tight junctions at the luminal side of the cells prevent the diffusion of sodium ions and chloride ions from the extracellular spaces into the lumen. This leaves a hypertonic solution close to the lumen, causing water to diffuse from the contents of the lumen. The electrolytes are absorbed by a variety of mechanisms, like those described for the small intestine. Essentially, there is a net movement of potassium and bicarbonates, that is, ions from the blood into the large intestine, because of the potential difference set up by the asymmetrical absorption of sodium ions and chloride ions across the cell wall. A question comes to mind: how brain play role in regulating the movements and functions of parts of the large intestine? The caecum and the ascending and transverse colon are innervated by parasympathetic branches of the vagus. The descending and sigmoid colon, rectum, and anal canal are innervated by parasympathetic branches of the pelvic nerves. From the sacral spinal cord, these parasympathetic fibers innervate the intramural plexuses. The sympathetic nerves via the superior mesenteric plexus and via the inferior mesenteric and the superior hypogastric plexuses innervate the proximal and distal parts of the large intestine, respectively. The rectum and anal canal are innervated via the inferior hypogastric plexus. Stimulation of the parasympathetic fibers causes segmental contraction, whereas stimulation of the sympathetic fibers stops the colonic activity. The internal and external anal sphincters usually keep the anal canal closed and are controlled both reflexively and voluntarily. The internal sphincter is made up of circular smooth muscle, and the more distal external sphincter is composed of striated muscle, which is innervated by motor fibers from the pudendal nerve. Making our way towards the end, let us take a look at gut microflora. Most of the bacteria that are present in the GI tract are found in the large intestine because the acid environment in the rest of the tract destroys most of the so-called microflora. 99% of the bacteria are anaerobic, and most are lost in the feces, which is said to contain 1,011 bacteria per gram. The bacteria are involved in the synthesis of vitamins K, B12, thiamine, and riboflavin, the breakdown of primary to secondary bile acids, and the conversion of bilirubin to non-pigmented metabolites, all of which are readily absorbed by the GI tract. The bacteria also break down cholesterol, some food additives, and drugs. So that was all. Stay tuned to sqadia.com and explore all such physiology-related medical content.